Genetics. So we'll start off with a couple of things. Again, one of the beauty with genetics and the majority of the SNPs, the single nucleotide polymorphisms that we start testing, is that most of them can be controlled through dietary changes, exercise changes, lifestyle changes, and supplements. So that gives us such a great area to play with. Some of the general ones that have to do with anxiety, the ADRA2B gene, or SNP. This codes for how much glucocorticoid production is made for each stressor. This is independent of whether that feedback loop is lost. This is independent of how many times during the day we call upon it. But it's for each stressor, how much of this do we make? Then there's the COMP gene. COMP gene controls how quickly we break down that adrenaline, noradrenaline, as well as cortisol. COMP gene also breaks down serotonin and dopamine and estrogen, and these genes will do other things as well, but a large portion of what they do is glucocorticoid control. With the ADRA2 gene, if we have that polymorphism for it, where we code for a DD, that means that that individual is producing more stress hormone for the same stressor than somebody who doesn't have that polymorphism SNP. AA for the COMT means that they're breaking down their adrenaline and noradrenaline very slowly. So now we have an individual who, independent of what that stressor is making more stress hormone, breaking it down and metabolizing that stress hormone less quickly. So those hormones stay flooding and surging through their brain and through their body for a longer period of time. These are the people that have higher panic attack levels, higher cardiovascular levels, and have been shown to have increased obesity levels. These people have also been shown to have a very, very small hippocampus. Hippocampus, again, is what is our short-term memory focus gland, and cortisol inhibits the production of the chemical messengers that bind into it, which is why when we're stressed we have sort of more racing thoughts, that beta wave activity, but we have a very difficult time focusing in on one specific area. These people have been shown to have hippocampus about a third the size of somebody who codes the opposite because of those flooding of glucocorticoids. What's even scarier though, and this is sort of like bordering on sci-fi almost, is glucocorticoids have now been shown, again particularly once that HPA feedback has been lost, to cross the nuclear membrane, bind into messenger RNA, change the expression of messenger RNA, and therefore DNA transcription. So it's actually not just turning on and off our genes, it's actually starting to change our genes. And that's a really, really scary thought, because once again, when glucocorticoids are doing this, it's never going to change that gene for a more relaxed, more healthy environment. It's always going to change that gene in terms of survival mode. So I'm either going to store more of my food as fat, I'm not going to let you fall asleep, because what if the saber-toothed tiger comes? I'm going to increase my heart rate so that I can pump out blood to my extremities a little bit more. 24/7, all of those have very adverse effects. What are some that specifically, SNPs specifically that have to do with metabolism? FTO. FTO has been sort of called our fat gene, and you know it's a little bit of a misnomer. There's at least a dozen genes that control what foods we crave, how quickly we break down sugars, how quickly we absorb fats through the intestines and put them into weight. Do we do better with a paleo type diet? Do we do better with a higher carb type diet? So there's so many different SNPs that we can take a look at, but FTO is one that is really well known, and it has to do with satiety. So yes, it will deal a little bit with the rate of metabolism, but it's more looking at leptin. And again, leptin is that hormone which at the end of the meal comes up, binds into the hypothalamus and says, great, I'm done, I'm satisfied, I don't need to keep searching out more food. So FTO, if you have at least one A allele, they're going to have an impaired leptin as well as an impaired adiponectin, the rate of fat burning. And these are the people that are really craving food, particularly from 6 p.m. on. Leptin should be most active in the evening because we're not supposed to want to eat after dinner. We're supposed to be sleeping. We're not supposed to be hungry. And these are the patients that you'll see that just can't stay away from the fridge after dinner. You're not hungry. You just ate 30 minutes ago, and they're back in those cupboards. It's like, what else can I eat? They might not even like what they're eating. They might actually even be full, but they're still back in there looking. And unless leptin sufficiently gets up to the brain, that message of I'm done and I'm satisfied will never be will never be produced, and they will always be driven back to the fridge. ApoA2, this is our body's response to dietary fats, both saturated and unsaturated. It's how quickly and how much of those fats are absorbed through the intestines, and then do they go over to the liver and go into LDL production, or do they go into white adipose tissue? Having just one G allele, and certainly if you're a GG, similarly with FTO if you're an AA, increases the amount of fat absorption and the amount of LDL production and the amount of WAT, or white adipose tissue, that is formed, even from the walnuts, even from the fish oils and the flaxes of the world. So as we'll get into this a little bit closer, just as Dr. Herkel was talking about curcumin with the one of the CYP phase 1 genes, down-regulating it, fatty acids, they're not for everybody either. We will start to see that some people are taking omega-3 fats, crossing the blood-brain barrier and forming amyloid plaques on the brain and greatly contributing towards Alzheimer's and dementia. So there isn't that one-size-fits-all anymore. So again, if we have a person here who's absorbing more of their fat than normal, decreasing their satiety signals, these are patients, again, that we have to start looking at leptin and adiponectin and dietary factors, and we'll get into all of that. Methylation. Methylation, as we know, it's basically a catalyst for absolutely everything in our body. Methylation SNPs were some of the first that were tested. Things like FUT2 and COMT has a methylation, one of the first genes we spoke about, MTHFR. But really, we cannot do anything in our bodies without methylation. And this was first seen as it relates to obesity. They took 25 obese individuals. They put them on a really low-calorie diet, and all they did was look at methylation rates of promoter ATP10A. Promoter ATP10A gene is what modulates body fat asymmetry and lipid transport through the body. What they found was those women who had the slowest amount of methylation of the promoter ATP10A gene, despite the fact that everybody was on a low-calorie diet, they lost the least amount of weight. Then they put everybody across the board on high-calorie diets. Those women who had the lowest levels of methylation, again, gained the most amount of weight. So we can see here, again, food's not our issue, it's methylation. And we know that cortisol has been shown to decrease methylation across the board on all genes. And one example bringing in that cortisol is they took 27 stressed obese women, and again, they did the same thing. They put them on low-calorie diets, and here they were looking at methylation rates of leptin and TNF-alpha. They did this also looking at 24-hour serum cortisol levels. What they found was those women who had the highest level of cortisol had the lowest level of methylation rates of both of the genes and lost the least amount of weight. Took the same group of people, put them on a high-calorie diet, repeated it. Those women who had the lowest level or highest levels of cortisol, lowest levels of methylation rate, gained the most amount of weight. So once again, it's not our food, but it's our brain and our stress hormones